Okay, so the way that it's going to work is, as you've already found, there is a chat box which will allow you to put any issues you've got with sound or video. If you put that in the chat box, myself and Duygu will be able to see that as we go throughout the event. There is a Q&A box where you can post your questions at any point. Please put your questions in at any point. If there's anything you want to know. We may wait until the end of the presentation to gather them up, because sometimes questions are on the same subject, or we can answer them at the same time. So we may not answer them straight away, but please post them whenever you feel like it. If there's anything you'd like to ask. Also, I may go in and answer it in text or with a link, because it may be more useful to do that. You can also post your questions anonymously. So if you don't want to put your name to a question, by all means do so anonymously. That's absolutely fine. What I'm going to do shortly is I'm going to hand over to Duygu. Duygu is going to give a presentation, and also I'll be doing a poll as we go throughout the event. I'll be asking you a couple of questions. If you could fill that in, it'd be really great to find out your responses to those. But I think that now we've probably got most people who've managed to join. I can see someone's got their hand up. If you've got any issue, just pop it in the chat box. Ask me a question. If I can help, I will. And just to let you know, we won't be activating anybody else's cameras or microphones throughout this, because there's a very large audience. It will become unwieldy and quite difficult to do it. It's also not possible that we could record it and share it if we then showed other people's faces and voices. So what I'm going to do now is I'm going to hand over to Duygu. I will be here in the background, even if I turn my camera off, just to save on bandwidth. So very warm welcome. Glad to have you all here. And I'm going to hand over to Duygu.
Thank you, Kim. I hope everyone can hear me okay. So if there's any issues with the sound, please let me know. Hello, everyone, and good afternoon, and welcome. So I'd like to welcome you all to our first taster lecture for this year, actually, which will be on digitalization of biomanufacturing. So I'm Dr. Duygu Dikicioglu, and I'm an associate professor in digital bioprocessing at the Department of Biochemical Engineering at UCL. So in today's lecture, we will be mainly looking at how data science can help us or assist biomanufacturing, and where digitalization of bioprocess industries in general falls within this broad domain. So before we dive into our subject, I just want to give you a very brief overview of the type of questions that I'm interested in addressing in my research. So primarily, I focus on the adaptation of any sort of digital technologies to develop data-driven and mechanistic modeling approaches in order for us to handle the complexity that we observe in microbial and mammalian biosystems and the bioprocesses associated with them. Now, I'm aware that this is a bit of a mouthful, but in practice, I use tools of systems engineering and systems biology, tools and principles from systems engineering and systems biology together to address basically the problems that we currently face in the design, in the development or the control of both cellular events and the bioprocesses that we actually rely on those cellular events on. The general idea behind this is that if we can integrate the concept of the cell being used as a factory with its cognitive bioprocess, then we can make well-informed decisions on how to improve our understanding and design the bioprocesses in a much better way to facilitate automation or to lead us to rapid process development and improved biomanufacturing practices. In my group, we also focus on developing different types of computational tools and pipelines for predicting and controlling system behavior as well. So these are all very high-level introductions to what I'm doing, but why does it matter at all, essentially? This is what we would like to know. So why would it be any important at all? Hopefully, in the following few slides, this will become more clear. Now, after this very brief introduction, I would like to actually It's quiz time, and I would like you to think about a question. So there won't be like an interactive quiz on this, but I know it sounds horrible, but I would like to just take a couple of seconds to think about the question that I'm posting on here or on this slide. So the question is, how many years do you think, in your opinion, would it take to develop a drug from the very beginning from the very early stages of discovery of that drug, all the way down to manufacturing and like getting the regulatory approvals and everything that needs to be in place so that it's marketed? So I would just like to, would it be a couple of years, 5 years? I see someone in the chat attended more than 10 years. Would it be 20 years? Like, yes, 3 years, 12, 5, 10. Okay, so this is, this is actually what one of the challenges that we are facing. So the slide I have here is an industry average that is acquired from the Association of the British Pharmaceutical Industry regarding the timelines of developing a drug. So as we see here, any development process starts generally with the selection of the relevant candidates from basically among sometimes thousands of options during the discovery phase, which would last approximately about 4.5 to 5 years, which would be expected to last. And any promising candidates are then carried over to the preclinical and the testing and the clinical trial stages. So this is a very tedious process, and less than 12% of all the products that enter the clinical trials actually can manage to make it to market. So if one potential drug, let's say, is lucky enough, it can make it to market in approximately 10 to 15 years. And it would also show us the timeline. The same timeline would be the same timeline for anyone who is expecting to benefit from that candidate drug or candidate product. So this is just one bioprocessing example to give you a very brief idea on some of the challenges that we are currently facing in the industry. Now, there's a substantial amount of effort that we actually place on not only on developing new products, but also on improving the process development to improve the product quality, and also to speed up the whole process to reduce the time it takes for us to be able to market that product, hence reduce the associated costs and render all biomanufactured products more accessible in the market. Now, data science has increasingly become one of the most powerful tools that we have available in our hands. But does this mean necessarily that actually mathematical modeling in bioprocess applications is a new concept to us? Not necessarily. We are engineers, and design and simulation has historically been pillars in developing new manufacturing technologies. So biomanufacturing has been no exception to that. And we have been utilizing mathematical model-based approaches in biomanufacturing for quite some time. However, while we are using those for quite some time, so what have we been doing, for example, within our department or within bioprocess industries in general? So we use, most of the time, we choose to use advanced simulation and optimization approaches to improve our product and understanding of the product and improving our process development strategies. We have been, for example, using, I hope you can see my pointer. Kim, can we? Okay. So we have been using molecular dynamics simulations for functional characterization of products. We have been optimizing metabolic pathways to improve cellular production capabilities, identifying and removing bottlenecks in the metabolic network, which would limit the cell's capacity or capability of producing more of the product. We have been optimizing process-specific operations, such as the initial stages here of upstream operation at the bioreactor stage. And we have been often utilizing very systematic approaches, such as design of experiments, to facilitate that optimization process. We have been designing biomanufacturing plants, conducting some economic analysis on these plants, and we have even been focusing on various different types of decisions around post-manufacturing stages, specifically looking at ways to facilitate the most efficient distribution of the specific bioproduct that we manufacture through its supply chain, for example. What changed? What changed? We have been using all these approaches, utilizing all these methods for years now. And what is now that we are talking about this digitalization of bioprocesses? Why are we putting now even more emphasis on the digitalization of, in general, process industries, and more specifically in this case, bioprocess industries? What's caused this to happen? So this has been the hot topic for the past couple of years, especially after the onset of the Industry 4.0 initiative. And there are several technological advances that we have experienced that triggered this change. Now, with the technological advances of the past 2 decades, we are now able to process and analyze samples at a much faster rate, any sample that we like, using superior techniques. And we can do this at a much higher throughput. And we can do this at an affordable cost. So we are capable of generating more information. Through the advances we have seen in computational infrastructure, we are now able, through the Internet of Things, for example, we are now able to link and allow the communication between different platforms and technologies. We are now, through cloud platforms, store this information at a much more efficient way, to store immense amounts of information at a much more efficient way. And through the advances in high-performance computing, we are also now able to basically process the data that we collect more rapidly and in a more accessible way. Now, in addition to those, we have also seen some regulatory components playing into this. And for example, the initiatives such as the Quality by Design initiative, or the regulatory agencies worldwide, reinforce the proper collection and storage of any data pertaining to the biomanufacturing of a wide range of products, including vaccines and biopharmaceuticals. So all of these, both regulatory and technological advances, actually pushed us to speed up the process towards making use of this data. So in light of these initiatives and the regulatory requirements that are faced by the majority of the bioindustry operations, we now have large amounts of data being generated, consequently being accumulating and stored by the minute. The question then becomes, can we make use of this immense and diverse data sets that are continuously growing to achieve some sort of a superior understanding of the biological entities that we use as cell factories, or cell lines and cell parts to produce our commodity products, or the manufacturing processes that actually allow us to market these products produced by these cell factories. So the real factory and the cell factory. Can we make use of this data to improve these processes. Specifically, what we are looking at is questions such as, can we shorten the time? Shorten the time for byproduct development? Can we render the products more accessible by reducing the costs? Or can we improve the manufacturing efficiency, or perhaps even suggest novel products or novel processes, novel ways of doing it, so that we would end up with a completely new technology or new way of actually new way of practicing. One thing at this point we might wish to ask ourselves is that, why would we actually need data science to achieve these goals? Can we just not keep on developing better and better versions of the models that we have been doing for the past few decades or so? So why do we need data science? Here, the primary answer that I have for this lies in the complexity of the systems that we are dealing with. Now, biological systems, due to their inherent nature, are prone to display some unexpected or random behavior, which in turn can lead to variabilities in the processes, at a more frequent rate than we would observe in other types of manufacturing or process operations. These uncertainties would, at the end of the day, necessitate making some decisions to ensure that we can dynamically adapt to changing environments and changing conditions. Therefore, we mostly take precautions to design the bioprocesses in such a way that we can maintain maximum robustness in the face of these uncertainties, hence the unknowns. Having access to information embedded in the large amounts of data that I've mentioned, collected from similar process operations, means that we are exposed to more of these uncertainties by the day, so that we are better equipped to model resilient systems and resilient processes, or take appropriate actions to counteract the impact of these random changes that are brought about on a day-to-day basis. So by now, I hope I have given you a very brief overview of why we need data science to assist us in improving biomanufacturing, and how this general concept came to fruition in light of the recent advances that we have experienced. Now, I would like to switch gears a little and focus primarily on our own efforts in the domain of digital bioprocessing at the Department of Biochemical Engineering here at UCL. Digitalization is a very wide, comprehensive umbrella term that embodies the use of digital technologies, essentially, and digitized data to impact how work, or in our case, research, gets done. So, I have tried to summarize some of the digital technologies that we use most commonly in the department, which help us address and solve problems in biomanufacturing, product and bioprocess development. We primarily focus our efforts around data analytics, machine learning, artificial intelligence, robotics and process automation, as well as on implementing data collection and storage standards. Now, we employ these technologies in shaping future biomanufacturing operations across a range of sectors. These sectors include biopharmaceuticals, cell and gene therapies, vaccines, industrial biotechnology, bioenergy, biorefining, bioremediation. Now, these different operations all face specific challenges, actually, that necessitate us to tailor the tools that fall within these digital technologies that I've actually briefly mentioned earlier. However, we can observe some common themes common problems that emerge, and there are some common methods that we can actually adopt to address these challenges. Now, we apply digital technologies to all stages of operation, from the development of the cells that we will be using in manufacturing to the development and optimization of the process itself that will be used in manufacturing, to monitoring and effectively controlling the manufacturing stage, to the assessment of the quality of the product, and to scenario modeling for the supply of the manufactured product itself. We employ data analytics and machine learning primarily to speed up the cell line development process. Now, this could be either a single, very specific single clone that we might be after, or a group of different species of cells coming together to do some concerted action. In either case, we make use of systematic use of machine learning and data analytics applications. We utilize the many measurements that we are now able to collect from our process operations to develop what are called the virtual, or for example, soft senses, to monitor and advise the process operation. We use exact in silico replicas of our operation to simulate possible outcomes to ensure smooth operation of the manufacturing using a technology called digital twins, for example. We capture novel insight from historical data generated on similar processes and implement data-driven control strategies to guide future processes, for example. We use AI-based methods in conjunction with molecular simulations to improve the quality attributes of the product that we are basically targeting to produce. We make decisions on what, for example, strategy to employ to enable therapies or treatments to patients or products, to consumers, or in any case, end users in general, in the most rapid and effective way. We evaluate options like whether a distributed or a clustered manufacturing capacity would work better in certain cases. This might even involve, for example, the evaluation of bedside manufacturing operations, as in the case of cell and gene therapies, and I'm sure this will be discussed in greater depth in the upcoming lectures that you will have in the following weeks. And last but not least, we also develop some strategies to standardize and automate process pipelines and workflows through robotic systems. Now, at the end of the day, all of these are engineering applications, and we are, as engineers, our dream world would be an optimized one, I'm sure. So our main goal would be basically to achieve an optimized process operation, which gives us the best functionality product in the most efficient way, in the highest efficiency. And we would like to basically shorten the time we take from the initial conceptual stages of that product formation to the market. And by specifically using the guidance we receive from data science. And consequently, we would like to reduce both development and manufacturing associated costs to make things more accessible. And we would like to ensure that we are doing it in a sustainable way at the same time. Now, this brings me to the end of my talk. I hope I have managed to give you some very brief idea about the very exciting work that we are carrying out in our department in this digital domain, and application of digital technologies to bioprocessing operations. Thank you for basically listening and joining us today, and I'm happy to take any questions you might have.
Great, thank you. So please put your questions in the Q&A box. We really look forward to going through them. And I'd just like to say that some of the things that Duygu touched on, things like cell and gene therapy, we have a session coming up on cell and gene therapy which will go into that in much more detail. Next week, we're going to vaccines. Again, we go into much more detail in vaccines. So if you really want to find out more about the particular issues and opportunities associated with those subjects, please post them there. And I'm going to get a link to the rest of the Spring into STEM series. But just whilst we're waiting for any questions to come through, I'm just going to ask Duygu, can you explain a little bit about what decisional tools are and what the kind of tools that you can use in this process and what they might be?
Most of the decisional tools that we have already been using for quite some time at the department are basically sourced from operations research applications in general. So anything you would be familiar with, like any sort of optimization tools, Monte Carlo techniques, or heuristic approaches that are employed in operations research, are applicable to us evaluating the bioprocess systems in general, in terms of basically guiding our decisions on any aspects of the specific manufacturing process, design of the process, or any other aspects of it that I have mentioned so far.
Cool.
There is one question in the chat. We simulate production of viral vectors as well. Simulate, I'm not entirely sure what you mean directly by simulate, but two members of the department, including myself, are focusing on technologies around viral vector product modeling systems that involve viral vector production. So if you have any more specific questions about what you mean by simulating the production, then perhaps I would be able to help much better. But in general, if it's the specific process operation that you're looking for, yes, we do have ongoing research on that.
Okay, somebody else has asked about practical training in manufacturing and cell and gene therapies. Again, I think if you'd like to register for the session with Kasim, we can go into a lot more detail about the lab-based work that we do there. And I'm just trying to think whether any of the other sessions we're doing, Duygu, is anything you'd like to add to that?
I mean, I wouldn't be the right person perhaps to address the practical training in cell and gene therapy domain, but can I just take the couple of questions that appear in the Q&A for the time being? So the first question I have is, the feature selection look like in biochemical engineering context? Okay, so I can give you an example on some of the recent work that I have been carrying out. For example, we were interested in antibody production operations using Chinese hamster cells, monoclonal antibody expression systems. And basically, we worked with quite a few data sets, historical data sets, and we were trying to understand whether, looking at it in a time series manner, can we predict what's going to happen at the end of the culture process from a much earlier time point. And in order to build those models, we needed to determine, so in the process, as I said, to basically to align with the quality by design initiatives and the regulatory, the regulatory aspects of manufacturing, especially during process development, product development, everything is very well recorded. So there are so many things being measured. And we have records of like 30, sometimes 35 different quality attributes and quantity attributes available to us per a single culture. And we have data on hundreds of those, for example. So not all of these are super useful in building these predictor models. So we need to process these features, such as like how much the cell grows, how much viability we can maintain over time, how much glucose it, for example, consumes all of a sudden. So all these things are actually things that have an impact on the outcome, or the things that have a potential impact on the outcome. So which ones would be the most important ones actually for us to develop or construct better predictive models? We use feature selection algorithms, for example, in order to basically address these questions. Through processing these different types of quality attributes, we can identify that, okay, if I'm interested in predicting the quantity of the product that I'm getting at the end of the day, and I'm building models towards that, I can see that this feature, this quality attribute, and this quality attribute actually play an important role in building models that predict that. Whereas another quality attribute, let's say I'm completely making things up, glutamine uptake does not matter all that much for the specific process. So if we can, at the end of the day, receive through the feature selection algorithms, we can receive some sort of information to guide our model building process for us to identify the exact features or the exact parameters that we would like to include in our models. I hope this explains your question. So the second one is optimization process. Oh, okay. So I am an engineer, but I have worked on the domain of systems biology for quite some time. So the question is, in case I'm not sure if everyone can see the questions, Kim, but so the question is, have optimization processes helped you uncover any fundamental biology? So I have been using metabolic modeling applications for quite some time now. So and metabolic modeling relies heavily on the concept of optimization. It's essentially what you're trying to solve is a linear programming problem, or depending on the type of questions that you're asking, it might be a quadratic programming problem too. But at the end of the day, using metabolic modeling approaches, I was specifically able to, for example, identify the cofactor requirements of enzymes in total within the cell. So this is not something that we can experimentally achieve at the moment. A prediction is as good as it will get. It can get or it will get. If we are looking at the total requirement for a specific cofactor of the whole metabolic network, this is only achievable. A prediction is only achievable by modeling. So, in terms of providing us some insight on biology, for example, this is probably something that could count as achieving novel insights through optimization. The next question is, are these techniques used in large-scale manufacturing? These techniques, meaning, so I'm assuming the techniques based on AI or specific digitalization techniques, or the ones that I have mentioned that we apply within the departments, like application of soft sensors, digital twins. These are all actually they have been demonstrated in large-scale manufacturing operations as well. So it's not only specific to related to by process optimization, small scale by process optimization operations, but also to large scale. In bringing these new computational approaches to reality, I think biggest challenge would be so the question is, what are the biggest challenges in scalable what are the biggest challenges in bringing these new computational approaches to reality? I think one of the biggest challenges that we can think of in this domain is to understand the strengths and the weaknesses of the tools that we are using, what they can achieve and what they cannot do. So we don't want to, so when something is a big hype, then there is always the risk of over-promising and under-delivering. There are some things that these tools are very strong in achieving, and we need to embrace the suitable tools to address the specific questions that can be addressed using those tools. I think understanding their limitations and understanding specifically where they are very strong at is the primary challenge, because if you use it for the wrong purpose, for anything, if you use it for the wrong purpose, you will say, it doesn't work. And all of a sudden, this big hype will become a boom. But what we would like to end up with is basically making the best use of these available tools in the best possible way to basically get the most out of them. So I think the biggest challenge is being very clear about what they can do and what they cannot do, so that they are used in the most suitable way. What are the biggest challenges in scale up of I mean, this is a very this is a very difficult question, I would say. There are quite a few challenges, but at this point, I think we are at this stage. We're not even in some cases thinking about not even scaling up, but scaling out. So that's I will give you one other thing to think about in that domain. So basically. For scaling up of processes, we now have some good tools in our hands to guide our process, and we are generally able to make those decisions. Like if we go for a scale up, then we know what to do, let's say. But in most instances, the bigger question might be perhaps to decide whether to scale up or scale out. And that's, I think, one of the very important aspects of the decision making that needs to be involved, and where data science can actively contribute quite a lot. And we have ongoing projects in the department, perhaps in relation to again, cell and gene therapies. Perhaps Kasim would also like to mention how these actually relate to cell and gene therapies. So I have briefly mentioned, for example, are we going to centralize manufacturing or bring it to, for example, as a bedside therapy, for example? These type of decisions will benefit greatly from the incorporation and utilization of data science. The next question we have is, how does the production of active pharmaceutical ingredients start relating to upstream processing? So I'm assuming that you're interested in knowing once we basically have our candidate clones available to us from the cell line development stage onwards. So then the next step would be basically to test the candidate clones to see whether they are happier in growing in certain environmental conditions, which will be used in the actual work process. So they have all their own tiny unique characteristics that actually we would like to account for, and therefore the growth environment or the growth conditions for these cells will have to be adjusted in order to improve the design space specifically tailored for the candidate clone or the clones that we're evaluating. So before that upstream processing, this upstream process development stage, first looks at how these clones would grow in different varying conditions at a very small scale. Ideally in, for example, if you are talking about biopharmaceuticals or something like antibody production process, we would start with 15 milliliter cultures and testing various different conditions. And then based on the information we get from them, then the operation, perhaps some of the clones will be actually not of interest anymore. And with the remaining couple of candidates, the process is taken to bench scale 3-liter, 5-liter cultivations to further fine tune the process operation conditions. And also evaluate the quality of the product that we are getting from that process. So it's a gradual line of scaling up towards the actual production stage. So the process development for upstream processing goes as follows. To what extent is modeling generally used in industries? In industries, I hope it's bio-industries that I mean, I will only focus on the bio-industries part at this point. I think there is quite a bit of a variation. So in terms of their research and development aspects, there is quite a large amount of interest and investment in this domain, because this is basically what the whole industry 4.0 initiative is about. So it's the automation of the process. It's the understanding of the process to reduce the reliance on the human decisions. Can we basically get a better understanding of the process through putting more emphasis on the digital space? So because of this initiative, there is strong interest. I don't think there is any specific application where all manufacturing automations are fully automated and decisions are made by AI. And I'm not sure if we would be able to reach there in a very short time frame anyway. But I think modeling especially is used in the product development and process development stages at this point. So not in the actual operation, but in the research and development stages of the process. Is model interpretability important? What would you say?
So if I could just quickly answer a question. Somebody's asking about when they can see a recording. It'll take me a couple of weeks to process it and upload it somewhere, but I'll send everyone a link afterwards letting you know where the recording is. Somebody else has asked about the background of PhD and postdocs. We'll organise events for postdocs at another time when we can go into way more detail than we can here. So if it's okay, I'll shelve those questions for another time, but you will get a link to this recording in the coming weeks. Okay, thanks, Duygu.
Okay, so after this brief advert, so is model interpretability important for your research? This is something that is a relevant point of discussion for data science in general. And I mean, from the interactions I have with computer scientists, I can say that in fact, even the machine learning and AI-based modeling approaches, which are actually more results focused than providing a mechanistic understanding of what the model tells us, hence the model interpretability, is actually moving towards a direction of incorporating that understanding, that results-focused understanding, with a mechanistic modeling approach. So these are the goal of actually many of the initiatives in the domain of machine learning research are now more inclined towards adopting more hybrid systems, so that we can actually use machine learning and AI, artificial intelligence, but at the same time be able to interpret the models themselves, as well as the results coming out of those models. So there is a strong focus and a strong shift of direction towards hybrid in order to facilitate this. What is the standard or acceptable range of model accuracy? Oh, it depends on the application. So there is no hardcore rules for that. Sometimes if the process has very little uncertainty in it, then you need to be very strict about the accuracy of your model. But if you're dealing with a high level of uncertainty and you would like to develop or construct a model that encapsulates basically pretty much everything that can happen out there, you need to account for that uncertainty, and you need to obviously relax your expectations from the accuracy of the model. But then consequently, you need to be aware of the system that you're working with, so that you can, when you're interpreting the results of your model, you're very careful about what the implications of those interpretations are. Have there been any real life industrial scale? Not that I'm aware of in data science use of or applications of data science in biorefinery. Not that I'm aware of. But I mean, if you have heard of anything from a newsletter or anywhere, I would be more than happy if you could share that. That would be quite an interesting domain. How close are we to achieving full autonomy? Okay, that would be biomedical manufacturing. So I wouldn't be the right person to address this question, because I mean my understanding. So I am more in the domain of process industries, but biomedical manufacturing, so the manufacturing of biomedical devices, this is a completely different domain and is not currently focused in the department either. So I think whatever I say would be very wrong, because I wouldn't be the person to talk about it. So I will apologize and not take that question, if that's okay. So I think I have gone through many of the questions, but I'm not sure about.
So just a quick thank you to everyone who participated in the polls. It's very interesting to see who's interested in postgraduate, undergraduate study, and those who have an offer to study with us, and those who don't yet have an offer to study with us. We look forward to your applying here. But it's also really interesting to see that there are quite a few people who have offers to study elsewhere at UCL and have come along to this taster lecture. I'd really like to say that if you're doing an MEng, so if you're doing the 4-year program at UCL, you can choose to do your final year in an entirely different department. So if you're in chemical engineering, you come and do a year with us. If you're in biochemical engineering, if you want to, you could go and do your final year with chemical engineering. There's a huge amount of flexibility in how you want to tailor your degree to your particular, what you find out you want to study as you go through. You've also got the option of minor modules, which means that you can tailor your degree to how you want your career to go. So if aspects of modeling, of data, are things that really appeal to you, you do have the opportunity when you're doing a degree to develop it to your own particular interest and really grow a career in a very exciting area. So we're really pleased that you're here and interested in this, and we hope that you take advantage of other taster lectures that are going on to find out what else is going on across engineering. I've put a link to the other taster lectures. I'm just asking if there are any questions that that we haven't answered, or anything any further clarification coming through. So we've got a few more minutes.
Can I just talk on to one of your comments? So if you're interested in an undergraduate degree, these minors that Kim mentioned, there are 2 specific minors if you're more interested in data science aspects. That actually both, one of them is basically hosted by our department, another one is hosted by computer science, that specifically focus on giving a better introduction to data science and how this can be employed basically in your own primary major or in a completely different application area. They are very exciting and very popular minors as well. I mean, from the numbers that we get. it's so exciting to see that there's like this interest and growing interest in multidisciplinary approaches being adopted, and UCL is a great place for that, actually, mainly because of even this whole concept of having minors and allowing students to explore some area, an additional area of interest for their for basically as an opportunity is great. Oh yes, okay. So you're sharing the poll results.
But thank you very much for joining. We hope you really enjoyed it. We hope you're having a great day, and we hope you enjoy any others that you attend. And if you have any questions which you didn't get the chance to ask, or there's anything which occurs to you afterwards, I will email you. You will have the opportunity to get back in touch with us. And please leave your details with UCL if you haven't done so already, so that we can let you know about any other events that are coming up. So thank you very much. Have a great afternoon, and all the best.
Have a nice day.
Thank you very much. Goodbye.
Bye.
Bye-bye.